All right, Jason, you're on.
Okay.
Welcome back.
Welcome back, yay!
Yeah, sorry, I'm just doing some microscope rearranging here. All right, so
Okay.
so back to the 2013 key. So this is the key that sort of changed the definition of Evelias and Sphecodogastra. And the first couplet is really to separate the traditional Sphecodogastra from everything else. And by traditional Sphecodogastra, we basically just mean species that are oenotherae specialists. They all have a modified scopa, which is dominantly what this character is about. Trying to get this in focus. All right, so I'm going to share on my other computer here. Hopefully. This is going to show. All right. A little bit off kilter here. So it's a little bit hard to see because of the white background. But I'm kind of looking right up here on the hind tibia.
Okay, we're not seeing.
Oh,
we're not seeing it yet.
I'm going to try to share that again. I don't know why that didn't share.
There we go.
Yeah.
Okay, you're in.
So I'm looking up here in the hind tibia, and it's
We
and
can't
it's
see
just
the high tibia well in that picture. There we go. That's better.
it's a little bit. The camera's got a different binocular vision, so I'm looking through the wrong eye. This is the camera, so.
You have the same software we do, and I don't know, there's different ways to approach this, but I usually am looking at the screen as I'm adjusting the specimen rather than trying to do it through the binoculars.
Good style.
I don't know if that's helpful or not.
It probably would be, but so here's the hind tibia here, and I think you can see that there's this sort of fairly straight, simple row of hairs on the underside. That's the typical character for these oenotherae specialists. They basically have like a rake of hairs, and they're, because of the way that oenotherae pollen is, it's connected by these threads, and so they rake the pollen off, and they get basically all the pollen at once. Whereas in a typical Lasioglossum, they have these sort of plumose hairs, these really branched hairs that kind of wrap together to make a little empty hollow space in each one.
Jason, can you point those out again with your?
Yep.
And can you tell us we know this is Sphecodogastra, but what species? Or is it Sphecodogastra sensillata?
So this happens to be oenotherae that I'm showing.
Uh-huh.
But this is going to be the character for all the Lasioglossum Sphecodogastra in the strict sense. So this is the common northeastern species. Kind of goes from Kansas to the Ontario, the northeast, but there's other species that occur in the Great Plains in the west. To say they're common would be not correct. but they're not rare. But because of they tend to fly very early in the morning or late at night, so you're unlikely to see them unless you happen to be collecting the right areas.
Rarely collected, perhaps.
Rarely collected, but probably not rare. Oenotherae is, let's see if I can get this character focus here. Oenothera is relatively it has a very sharp perpodial carina and fairly coarse kind of rugae on the metapostnotum, and it's like a very black beam. So this is the only Sphecodogastra in the strict sense that has these big sharp carina that really stands out. And again, it's fairly eastern bee. So this is how you sort of key that out fairly easily. So if I jump back to the key, I can. The scopa with a single row of hairs jumps us to couplet 18. And here's sort of the image of that. Can you guys see that? It's not shared yet.
Nope.
Not yet.
Try again. Okay.
Yes.
So this is the image in the key, that those sort of scopal hairs. And that takes us to couplet 18. What's the first couplet in the key? I skipped it. And then we get to this part here where you either have a pale orange or black metazoma. And the ones that have pale orange metazoma have really enormous ocelli. I can show that in a second. And then you get to this couplet 19, which has this propodium carinate, which gives you Lasioglossum oenotherae. This is only for the east, and so we don't cover all of the western ones. But they would run till Lusorium in the eastern key. But if you want to key those out, you would use McGinley 2003, and then you would be able
How
to
many?
Sorry?
How many species total in that traditional Sphecodogastra group?
I would
So
say
how many?
maybe 8.
Okay.
There's so there is the two with enormous ocelli and red abdomens that would key out to texanum here, and then there's all the other ones are entirely black. There's Oenothera in the east. Lusorium just barely gets into the east, so you can sort of maybe get it in like Minnesota, Wisconsin area. But Lusoria is probably one of the most widespread in the West, and it looks very much the same as aberrans. And then there's one that's kind of restricted to California, Antiochiensis, and then there's a couple that are kind of down in Texas, Danforthi and Potosi. But they're very recognizable, but you have to sort of key them out using McGinley 2002.
And they all have slightly expanded ocelli, right? Although some are quite huge.
Yeah, it's the ocelli are the expanded ocelli are kind of hard to see in the black ones, but they're really obvious in the red ones. Let's see if I can.
Meaning that they're just larger in the red ones, if I recall.
I'll see if I can. See if this can pop
Yeah,
up.
it's up.
Yeah, so this is actually Nocta Vagum, which is the one that's not in the 2013 key, but it's So it's kind of in the kind of Great Plains, Texas kind of area. But you can see a little bit obscure, but you see the size of the ocelli, the lateral ocelli here. And the median ocellus is kind of behind the scape, but they're just enormous. They're ridiculous. Kind of similar to like Megalopta in Central America. It's really big. These fly at like full moons at night, really dim light. Whereas something like oenotherae, I remember the first time I caught it was I got up especially early to go to work. So it was like 6:30 in the morning, and they were just on the evening primrose at one of my neighbor's house. So they're kind of early morning flyers.
And also late in the afternoon. I've seen them on here. Yeah.
I've caught oenotherae like 5 p.m. on a Rudbeckia plant once. Right. But it was a very shady street. Yeah. So
Yeah. This is the problem with a lot of floral records is if you wait long enough, every bee will be on every flower at some point.
So this is the abdomen. So a little bit out of focus maybe, but
Species are we
so you get the
currently
idea of why they're called Sphecodogastra, because they have sort of red abdomens, kind of orange.
So Jason, a question, there was that discussion on the ECN listserv, I think it was recently about, I think Joel was involved there, about red abdomen Lasioglossum diolictus group often having a dark spot on T2 and elsewhere associated with perhaps a spiracle. And then there was some not controversy, but some conversation around whether that was glandular or not. Does that occur on these Sphecodogastra that have red abdomens? I see some dark spots, but I'm not sure if that's just a blemish or is one of these modifications.
Your guess is as good as mine. Not really. Don't really see anything obvious.
Yeah. Okay.
and what species is still on deck?
This is
Texanum. This is Noctivagum, actually.
Okay.
Noctivagum has a slightly different head shape, mandibles, than Texanum. I think this is Texanum. The Noctivagum, the head is a little bit wider towards the base and it has kind of bigger kind of mandibles. Whereas Texanum looks a little bit different. A little bit narrower towards the compound eyes kind of converge a little bit more towards the lower part of the face.
Jason, am I tricking myself or are there longer hairs at the, let's call it the edge of the clypeus extending out towards the mandibles than in other Lasioglossums, or is that just something I'm making up a story about?
Well, you're not necessarily making up a story, but that's not a character that I have specifically looked at. So there are definitely this long setae that come off of like that kind of marginal part of the clypeus, but how relatively long they are, I don't know. I'd have to look. So I'm not sure if that's a character or not. Well, that's not the Vagum. Potentially show you a Texanum just for comparison. Get the head orientation.
While you're getting that ready, I'll share my screen and show the ranges here of two of the eastern species, if that's okay.
Well, I got, yeah, okay.
Oh,
No,
sorry.
go ahead.
Did I just cut you
No,
off?
you're fine.
Okay.
We're fine. We'll jump back.
Okay. All right. So here is the range of oenotherae, and here's the texanum, or is it texana? I think it's texanum. Oenotherae and texanum over here. And what's the other one? Is Lasaurum?
Lusorium?
So,
Yeah.
and so Lusci.
Oh, no, it's Orium.
And this is one of the advantages here in like using the Discover Life, the mapper itself, you can put multiple species on here. So yeah, so you can see how mostly there's I guess Wisconsin, that's probably John Asher's record of like a placeholder, and then mostly Western. So yeah, Prairie, Western, and Eastern. And also points out that we could really use more people's dots on those maps because a lot of information is not being expressed here, but that's a long ago story. Okay, Jason, go ahead, take over.
Okay, I'll just show you the texanum face for comparison. So again, really huge ocelli, super obvious.
Okay, you have to share it. Take over.
Sure. I can see it on my screen.
Oh, okay. Sorry. Yeah, I was on my computer, not on the browser.
So again, this is really big ocelli. It's got the kind of pale orange metazoma, but the eyes are a little bit more convergent towards the below, and the mandibles are not quite as enlarged as in Noctavagum. But Noctavagum kind of overlaps a lot with texanum in kind of the central part of its range, but doesn't occur as far east to make it into the eastern. But
am I missing it? But I still don't see your picture.
I mean, I'm on two different computers, so I can see it on my other computer. So
Yeah, we can see it, Sam. I can see it.
Oh, okay. All right, then I must be Oh, I see. I'm still theoretically sharing too, so never mind. I just turned that off.
okay. So that's texanum. Let's maybe show a Lusorium just for, I don't know what I'm going to show about it. I'm not going to show a Lusorium because it's more. Okay. I'm going to share. I'm going to go back to the key gradually. Realize I have to share it twice. Okay. All right. So we're going to go back to the beginning of the key. So everything else kind of runs to the normal scopa. And the first species we get rid of is something called Nellium bonus. It's kind of an unusual bee. It's, as the name suggests, it's kind of related to like marshy areas. Nelumbo is water lily, but I've never actually seen any strong evidence that it's actually a oligoid on that.
It seems to be a wetland species for some reason,
Yeah.
but not necessarily only on water lilies.
Right. Okay, so it's a really distinctive kind of bee. So I'm kind of towards the anterior margin of the sputum, the punctures get really dense and kind of rugose. So they're not, these distinct punctures like you see in most Lasioglossum. But the other sort of really obvious thing about this bee is the way the propodium is. And it's got this really clearly sort of delineated kind of propodial triangle. And then on the sort of kind of exterior part, what I call these sort of triangular areas here that I call the dorsolateral slopes, and kind of posterior, it has this kind of dense, oppressed, highly plumose hairs, tomentum, that you can kind of see.
For microscope, it is not available.
What's that?
We can't see, or Claire
no.
and I both can't see the picture.
Oh, sorry. Sometimes whenever I'm sharing, I have to share twice once to get out and once to
Oh, to flip them. Mhm.
flip. Sorry. So
Here we go.
I was talking like people were looking at what I could see, but so here is Snellium bonus. So again, so the anterior margin of the scutum, the punctures become kind of rugose, kind of this more like a wrinkling rather than punctation. And then this very clearly delineated propodial triangle, which and then all the propodium exterior to that has this kind of dense tomentum. It's kind of sort of short, oppressed hairs.
Yeah.
So this
and
is
very gray
bonus.
and very gray hair. It's not
Yeah.
bright white.
So in the East, there's nothing else like this. This is a very unique species in the East. Hard to mistake for anything else. When you go further west, you'll start to see some other bees that have a propodium kind of similar to this, not maybe as extreme. From Texas down to sort of Southern California, there's some various species that sort of seem to be very closely related to Snellium bonus, but are different. So Robertsoni is one, and Gustius is another, which is in Colorado, and then there's some undescribed species. But they tend to have more sort of distinct punctation, kind of longer heads than Snellium bonus. Snellium bonus has a pretty standard head.
Are they wetland related at all?
Not to my knowledge, but they're very rare. So we only have like a few specimens that just have like a town name kind of location data. So we don't have specific habitat data. Okay, I'm gonna jump back to the key. Let's do this.
That was Snellium bonus, right? So from character 2.
Yeah. So yeah, Snellium bonus is a species that it's good to find. It's not like the most rare thing in the world, but because it seems to be a bit of a habitat specialist, it's not. I'm always happy when I see it. Okay. So the other sort of half of that couplet is just basically regular punctation on the scutum, and doesn't have these sort of dense hairs. So that gives us to 3. Did you guys cover pectorale last time? Yeah.
Can't remember. So
I kind of feel not.
I don't think so. I don't have it highlighted, so I don't think so.
Well, pectorale is actually relatively abundant in some areas, and its kind of defining feature is that it's like got a really rugose or sculpturing. So if you're thinking in like Vander dialectus, I don't know if this is going to show up very well, but it's kind of so the character is basically mesopsternum entirely coarsely rugose, and it's kind of like if you took like something like a Cressonii and made it entirely black. That's kind of what you're looking at. So here's the mesia. It's kind of upside down, but
Share screen.
Screen
Yes.
problem.
Oh, yeah, sorry. Need like a reminder every single time.
There we go.
There we go. So, if you look at the mesopisternum, which is kind of visible here, this is just like completely coarse wrinkles, rugose. Not a lot of these species in this key are kind of weakly rugulose. They have some fine wrinkles, but it doesn't stand up. This is like a raisin, just like really rugose. And punctures are fairly coarse on the scutum, and the propodium, which may be visible in this specimen, is also very coarsely sculptured. So it kind of wreaks havoc with trying to distinguish the subgenera, because usually the Sphecodogastra, we talk about them having, like in a broad sense, we talk about them having carinate propodia, but the hemihelictus do not. So I'm gonna try to find you. So you can sort of see there's really coarse sculpturing on the propodium, and the carina don't go straight up like in a Sphecodogastra, really. They're kind of like out to the side of it. But then there's like these really strong oblique carina that kind of come across here. You can see my mouse. So it gives this appearance of really coarse sculpturing. And
Hey, Jason,
yeah.
do you want to maybe mention in a very broad way? So, for example, the male pectorale and the female pectorale look very close to one another, and other groups of these dark ones do not. What's the pattern?
So in a lot of the traditional hemihelictus, the males have relatively short flagellum ears, so their antennae do not look very long. So should be a male.
Which is often the case in like Dialectus group. Let's just in contrast, you can spot them a mile away, and it's often every once in a while I'm like, oh wait a minute, this is a male with pectorale, for example. But I think also Nellum bonus.
Yeah, at least if I have a male here, I can show you. Oh, I'll have a male here in a second. Let me see.
Okay.
Pectorale.
And then other groups develop a lot of yellow on the rim of the clypeus.
The problem with these is it's hard to find the males because they don't stand out. I don't know.
You can pull some up from Discover Life. I just put a link in the chat.
But I'm having trouble finding a male pectorale, but I can find.
Jason, Claire just put a link to a male in the chat for people if you can't find one. But I think the key thing is like it's super similar, and the scapes often are a little shorter, as is often the case. But because it's got shorter, on average, segments than say some of the other Lasioglossum groups, they can trick you up.
Okay, this is not a pectorale. I couldn't find a pectorale, but I found a lustrans, which I think this is a male. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Yeah. So this is a male lustrans.
So people should be aware that if Jason can't eyeball it and say it's a male or female, it's going to be tricky. So you got to watch out.
Yeah, so this is kind of true in a lot of like most of the Hemihelictis. The Foxii group is an exception to this, but usually the male antennae, it has 11 flagellomeres, but the flagellomeres are relatively short, so it has this sort of female-like appearance. And in a few of these, like pectorali, Ignelium bonus, right, lustrans, the faces are also entirely black, and so you don't have a lot to go on. Basically, just without spotting them by eye is a little bit tricky.
Right.
And
They don't necessarily slim down as much as, say, again, the Dialectus group males do. So they're still pretty robust, even on the abdomen.
Yeah. Yeah. So you have to look closely like this for males. But there are some, like I think you might have covered things like Pinky last time. That group, so pink eye,
Yep.
swank eye, and Federense, they also have relatively short antennae, but their males have like kind of these sort of macrocephalic heads. So they really have lots of yellow and big heads, so they stand out a little bit, obviously. I'm going to jump back to the key to see where we are, and then figure out what to look at next. If anyone has any questions, feel free to stop me, but okay. So we're getting into some other sort of rare-ish kind of stuff. So the next couplet is the pronotum has this sort of sharp angle that sort of extends kind of from the dorsal lateral angle down across the side of the head. This can be a hard one to sort of visualize. But we'll see if I can find these specimen.
It's similar to Andrina. So when when we talk Sphecodes and a couple other species where there is, for whatever reason, this raised, often very sharp, and sometimes that's an indication of species differences in running down the pronotal collar from the pronotal angle.
Yeah.
Pardon, Claire?
Swanky.
Swanky would be my
I'm going to see if I can. This is visible. Let me flip this.
When you see that sharp angle in any of these Lasioglossums, or it's a air quote good species. Like, oh, this is interesting. You'll have to figure it out from there. But the most common case for the most common species are the pronotal collar is a smooth horse collar type of thing where there's no really defining ridge or angle, as Jason is pointing out. Same thing would be true of the parasitic Dialectus species. Well, most of them, right? I think in a few odds and ends, like Heterognathan.
I'm not sure how it's a hard thing to show in 2 dimensions. Like this is something I want to be able to rotate the specimen around, because then you can see the transition of where the light is shining, reflecting off your B to where it goes black. And so it's easy to see. But you're basically looking right here. So you see this really bold kind of hot spot where the light is. And then there's this kind of edge right here where it goes straight to black. That's where you're looking. And if it's a rounded specimen, then that transition of light to dark should just kind of be gradual, because the light is just kind of going to bounce off one surface and then gradually sort of decay as it kind of goes around the bend. Whereas when that carina is there, it's just a sharp delineation. That's kind of what
Can
we're
you
looking for.
Can you sharpen that a smidge?
I'm trying a little bit, but
Yeah.
feel like, oh, maybe I'm not live here. Can you try that? Piece of dust just flew into my bead. That red
Wow,
string
look at that.
appeared out of nowhere. Hydrostatic forces. Yeah. So it's hard. Yeah, again, it's kind of hard to, but it's, I'm gonna put vertically because my brain works this way, but right here where my mouse is, that's an edge.
Hmm. Yeah, this is always a tough one for people starting out, is to see that. It's a great character, but it's one of those things where working with known specimens is particularly useful rather than trying to make up the presence of a sharp edge when none exists, as when you're starting out.
Yeah, and it's kind of independent. So so if you use like the Lasioglossum key, like McGinley 1986, it's a character that pops up periodically for certain species, and it occurs in our key for that. All of them in the 2013 keys are like hemihelictus. None of these Sphecodogastra have this character, and it's also something that you see in the parasitic Dialectus, especially. It seems to be connected to macrocephaly. So in the hemihelictus, all of the species, I think it's fair to say, that have this character in the female have macrocephalic males. And in the inverse, if you're looking at Dialectus, the way to find parasitic Dialectus males is that they have this character. So their heads are not macrocephalic, but it carries over from the opposite sex. So I think that's what's going on here. This is a carryover from the fact that males have this. Okay. I'm sorry about that little red string. It made my specimen look less attractive.
Are you wearing red?
No, it just flew in. It's even like enclosed in a little like diffuser, so it just appeared out of nowhere. Okay, so this is good because we're at couplet 4, so we jumped. It's got a sharp corona, so we're going to jump to couplet 5, and then inner spur with approximately 15 to 20 teeth. Supraclavicular sparsely punctate. And there's some other things that aren't in this key. So this is actually the species we have in front of us now. This is Lasioglossum pectinatum, which is a relatively uncommon bee, kind of mid latitudes, and it's a specialist on physalis. So if you have any ground cherry blooming anywhere near you, go look at it because it's got 2 bees. You can kind of see here, maybe slightly on the mesopisternum, it's quite smooth. You can kind of see some.
Need a share there.
Yeah, sorry, I'm. Okay. So it's a relatively smooth bee. I'll see if I can show you the spurs. The spurs are pretty hard to see.
So I'm unclear on what the last bee was. I thought it was Swanky or Swanky, but was it?
We are looking at pectinatum.
No, before that.
We had
No.
pectorale immediately before this.
So this is pectinatum. It's the same as last time
Okay.
because it has the sharp carina too.
I'll try to show you the spurs, but it still might be a little tricky.
Yeah, so those spurs are a great character. And a lot of times, though, I get very frustrated, particularly if you have the usual crumpled up specimen where the legs fold in next to the body or against each other. Sometimes it's hard to see that inner tibial spur very clearly. It's
Yeah,
not
I'm
a,
gonna struggle here.
and you may not have even enough magnification on that microscope.
Close, but I'm not there yet. Yeah, it's most Lasioglossum like Dialectus and uh, any Halictus tend to have pectinate intermediate spurs, so the
So you can really see big teeth, essentially.
so yeah, so normally you have these sort of, you know, the first tooth is really big and then it gets a little bit smaller and a little smaller, and you have like 3 or 4 of these almost like little fingers. Whereas this, there's a little bit out of focus, but there's just these little tiny fine teeth in that inner spur. So it's quite different than anything else that you'll see in an eastern Lasioglossum. So pretty distinctive. It's a very recognizable bee if you find it, but they're pretty rare.
Yeah, we've done a lot of collecting on Physalis and grow it here at the lab, and we've yet to come across that species, even though it is known from the state.
See if I can get in the face here. Got a relatively kind of slightly elongate face, but it's a very smooth, kind of shiny bee. Yeah, pretty distinctive. Yeah, it was Ty Rulson was the one who keyed in that it was maybe a Physalis specialist. And I was at Cornell, and I just first time I found Physalis growing, sure enough, he was on it. So I think it's pretty reliable. This kind of the range of this bee is not very well understood in that it kind of goes. If you look at the 2013 papers, this is kind of like straight line that kind of goes from like mid latitudes to kind of towards Illinois. Just kind of like it's just east west. There's like no north south kind of spread to it. It gets into Minnesota. That's about as far as it goes, as far as we know. So more people need to get out and look at Physalis.
Yeah,
If you don't get
Physalis.
Pectinatum, you'll get some Calides and Perdita that are interesting.
Right. And Physalis is very weedy almost in lots of circumstances. So it's around, and the range is super extensive.
Yeah, I think for me at least, my challenge with that is because the flower is kind of this kind of greenish yellow and it's upside down, that it's
Yeah.
easy to walk by it and not notice it.
Yeah, that's also true.
But it's worth looking at if you find it. Okay. Yeah, there's other bees that specialize in it too. Okay, so the standard state is that you have the metatibial inner spur that has like 4 or 5 teeth, and you know the one closest to the bee, proximal, is much longer than the width of the spur itself. So that's where we normally go. I think you guys covered Sopinki last time, so I'm going to jump past that. The other ones that we have. So Swanki and Fedorensi tend to be they're almost right. This is a swank eye. I'm still not sharing. That's right. Share once, share twice. So swank eye sometimes has like an orange metazoma. And the ocelli are maybe a little bit expanded, and they tend to fly kind of later in the day. And so they're kind of moving toward, it seems like they're moving towards like the Sphecodogastra in the strict sense kind of behavior. But they have normal scopa. If you can sort of see that scopa up here, this is a plumose normal standard scopa. So they don't have those rake-like hairs. It'd be really easy to mistake these for like a Lusorium style beak, especially whenever they have like a brown. That is it. The first time I saw a swank eye, I thought I had found some undescribed species of Sphecodogastra.
We have a male or female on deck,
What's that?
or I guess scopa, so it's female.
Yeah,
Female.
Yep, this is a female.
You get dark forms and the yellow brown forms, like in Sopinsi, you do. But is that true for Swanky?
Yeah, swank eye has brown and red forms. So let's see. This is the male. A little bit out of focus. And humans making sounds. Okay. So unlike some of the other hemihelicus, it's got really bright yellow mark, like Clypeus. Bright, and the mandibles are quite long. They kind of extend quite far, and the Clypeus is quite flat. So it's kind of almost like a state you see in some of the Lasioglossum males, the subgenus Lasioglossum. And this is a character. And the genus is a little bit wide, but this is kind of this kind of face shape, the clypeal mark, the big kind of like pointy mandibles is similar in males of Slopinki, males of Fedorensi. It's kind of like they're a unit. They're kind of a closely related group of species. Don't know if I have.
Can you explain so this is some obscure language in the key, hypoemperal area punctate, and show the pronotum with the carina ending posterior of the oblique sulcus.
Yeah. I'm gonna see if I can show you something along those lines, but that's a bit subtle. The male, it's a bitch. It's a male.
Doesn't like Mitchell give that a different name?
I think Bradley maybe describes it. See if I can get this in focus for you folks.
It's basically a mound just below the forewing on the mesepisternum or the pleura.
I don't think this is in the key, but there's another way of telling apart Schwenki and Federensi. This is a Federensi, and the length of the propodium, kind of the longitudinal length, it's quite short.
Oh, yeah.
Distinctly shorter than the scutellum. Whereas in a Schwenki, it's longer. It's more similar to the length of the scutellum. I've never seen a Federensi with a red metasoma, although I wouldn't put it past them. So originally I thought, oh, Schwenki has got a red metasoma and Federensi is black. But there are black Schwenki. But they're not as common as the red form as far as I can tell. It would be very hard to show you the pronotal character that's in the key. Let's see. I'll jump back to the key just to see if there's anything else to go off of.
And
Define the hyperemeral.
hyperemeron. And also, just a quick question too, Jason, is are all these three Sopinsi, Federale, and Schwenki, are they all sand specialists?
As far as I know, so I think one of the longest series that I saw working on this paper was from the Indiana Lakeshore Sand Dune National Park or whatever it's called. We get Schwenki here in Manitoba, but in our internal sand dunes that we see up here. Federensi, so Schwenki seems to extend a little bit farther north. It kind of goes up into like Alberta even. And Federensi seems to be a little bit more localized in the Great Plains. Schwenki is.
Yeah,
Federensi is from Fedor Tex.
Yeah, I've only seen these in sand areas.
Yeah. Yeah. And yeah, so they can be abundant if you find the right location, but generally speaking, you don't see them in collections because they have restricted habitats and they seem to like to fly kind of outside of normal working hours to some extent.
Yeah, they will show up. They will show up in bowl surveys, though.
Yeah. Oh, I would say, going back to pectinatum for a second, pectinatum males have a very extremely weird macrocephalic heads, but they're incredibly rare collections. So I sort of suspect that they're probably hanging out in the nests. We see them very rarely. I've seen maybe 2 or 3 in my life.
So Claire says there's a comment about in the comment sections, or she's interested in the defining better the what is it, the hypomeron, or I always get that hypoepimeral area.
Yeah. I'll see if I can find a specimen where it's not blocked by a leg or a wing.
Can you give us words also to define it? Is this like the anterior part of the mesopisternum or question mark?
It's like a little hill below the junction of the wing and the pleura.
Oh, that's what you're saying.
You're looking for. Let's see if I can share. Okay.
This is also a useful character in Sphecodes.
Okay, so this is the mesopisternum here. Here's your tegula. I hope everyone can see that. There is this preepisternal groove that runs down. You can sort of see this sort of hairs. That's basically where it is. That's a pre-episteral groove that runs down here. And in the back, you have the meta-episterum. That's this kind of plate here. It's kind of narrow thing that runs down to the hind coxa. There's the mid coxa. And right, you see my mouse?
But
Right.
yes.
Yeah, right where my mouse is, that's a structure called the scrobe. So it's kind of this little like pit indentation area. And there's a scrobal groove that kind of runs front to back along that. And this little box right here, that's the hypoepimeral area. So hypo means above, epimeron is this structure. So it's the top part of that. So right below the tegula, there's a sort of square. It's kind of defined below by the scrobal groove.
And
That's
it's almost
where-
always raised.
Yeah, it's because of this. Yeah, because there's a groove below it. It's kind of like a mound. It's it's defined in the back by this the separation between the mesoepisterum and the metaepisterum, and there's the preepisternal groove that runs in front of it, scrotal groove below it, and so it's got like a ditch of like a all the way around it. So it's kind of like raised area. But yeah, the sculpturing here sometimes differs from the sculpturing on the rest of the mesoepisternum. So like in some of the Sphecodes, like Sphecodes levis and Townsii, it's relatively smooth. And then in others, it's kind of wrinkled. And in Swanki, can't really see it very well here, but it's relatively smooth, distinct punctures, I guess. That's what the key says. So it's not a character that I use too often for Lasioglossum, but occasionally it's useful. So that's the hypoepimeral area. There's the pronotal lobe here. Orient yourself. So this is the top part of the mesoepisterum.
Well, we are at 2:00. Do we want to wrap it up here, or were there any more questions, or did anyone want to see Federenzi again so we can finish out that couplet? Going once, going twice. Hong Mei wants to share that they're hiring a postdoc to work on pollinator ecology.
That's crazy. Who is it?
Hi, yeah, it's me. So I work at Central State University. We are HBCU 1819 land grant. We got this USDA funding to hire a postdoc. The postdoc will work on the silflowers, silphium, and also all the pollinators come to silphium. So there's part of it is to all the bee identifications. If any of you know any good, PhD students graduating or anybody look for postdocs, please let them know. We have the job hasn't posted yet, but I have a link to a Google form, so somebody can fill out the form, and when we have the job posting ready, I'll just send to them. Is that
So,
yeah?
Just a quick question. Why silphium? Why, that's kind of narrow.
Yeah, so silphium, it's in a kind of like a sunflower type, but smaller. And we're collaborating with Ebony Morel from the Land Institute in Kansas. So they are doing a lot of breeding, cultivate of silphium. And we're trying to introduce this plant to Ohio for soil conservation, for pollinator health, for, try to have more habitat, good pollinator habitat for the land here in Ohio.
It is they can be very aggressive or thuggish. And I recently read a paper, I think I have this right, that they introduced one of the Silphium species to Poland, I think it was, as a biomass plant, which
Yeah,
is
so right.
Go ahead.
And there's also with the Land Institute, they have Silphium studies in Argentina, I think. I'm trying to remember South America, in Kansas, variety places. So we just wrote this proposal and we thought we'll give a try. And the first time we wrote it and we got it. So
Wow.
Nice.
yeah, so we are working with 10 different farms now. They have different design on the plants and pollinator population, and we want to compare them. Yeah,
Yeah.
and also study the special honey out of the bees.
Yeah,
Just
that'll be
comparison
super
of-
interesting.
Yeah, among different bee pollinators, honeybees and other wild bees. So I post a link in there. Anybody that's interested, or tell anyone you know. So we try to hire a good person to work on this project. It's a 3-year project.
Great.
Wow.
Thank you so much.
And Sam, do you want to mention recruiting experts?
Yeah, so Claire and I had a discussion today, particularly, we're coming into the holidays, so it's we tend to like, what are we going to talk about today? And then let's call Jason or Mike Arduzer or whatever. So we thought that we would try and schedule things out. And amidst that, we assembled a list of taxonomists. I don't know, Claire, did you post that at all?
I did not.
Okay. Who we're going to get in contact in addition to Jason and several of the other people. So if anyone has any particular taxonomists they would like to recommend or would like to hear from, we'll talk to them. So, we just put that list together, but we're going to do things like see if we can get Mike Engel to talk about bees in amber, get Molly Reitmeyer back on the talk show circuit to talk about Triepulus, Tom Ornifurco on Epiolus, Zach Portman on Macroterra and Perdita and other people. So we're just looking for a diversity of experts and or people with lots of experience identifying bees. So we're open to all kinds of stuff. We have a longer list. It's not just those folks, Terry Griswold, Corey Sheffield, Lawrence Packer. And we're not just restricting ourselves to the East. We're very interested because there are a lot of people interested in Western species too, to bring that in. So you can email us or you can drop it in the chat who you would like to see or what groups you're interested in that we haven't covered very much. We still have Nomata, but my collection of Nomata is at the Smithsonian right now. So we'll try and bring that back. And then I would love to spend 5 weeks at least on Nomata. It's my favorite group. So,
We'll just have one class per species.
right. Well, whatever a species is, like the problem with Nomata, you get the bidentate group and the molecular stuff is all over the map. And same is true of the white CT things like Cressonia, Pygmy, SAI, Illinoense. What are those? Is a really good question. And a lot of things in the past were color pattern oriented, like, oh, this has 6 spots on the abdomen and this has 8. So let's call them something different. And the molecular stuff for some of those groups does not behave well. Anyway, so a delightful, mysterious group. And there's a lot of rarity and interesting things too. Jason's been involved in several uncommon Nomata things.
Causing chaos.
Yeah. And then you go out west and it's like a whole different ball game of undescribed species. Anyway, that's what I got. Thank you, Jason.
You're welcome. I hope it was helpful.
Yeah, It definitely
Always.
is. And people are, we're talking to people and people use the online recordings all the time. So it's a good reference deck for people beginning because a lot of people can't necessarily attend live.
I'm behind on uploads and I swear I've gotten an email every day.
Okay.
Yay!
All right. Thanks, everyone. Bye now.